Yeah. She's still there. I retired. I wanted to get
welcome everybody. Thank you for attending the ACS San Diego chapters. I'll get a little bit quieter. It's okay. The San Diego chapter speaker series meeting. It's wonderful to see people here. I know this weather was a little bit intimidating, but it's nice to see the faces and all of you online. Before I present our speaker, I would like to share a little bit of information with you. Really, the first thing I want to do is recognize and thank the Legacy and her crew for allowing us to hold our meetings here. Yeah. It's very gracious of them, and we truly appreciate it. And speaking of boats, there's whales out there. We're seeing whales. So if you have not been out on the water lately, get out there. And one way to help you get out there is at our raffle prize today. One of our raffle prizes will be some free tickets on the Legacy. So anyway, get out there and enjoy that water. And then I want to share something that's maybe not as exciting, but important. So I'm going to kind of read this, if you don't mind. Today, we find ourselves in a very chaotic and destabilizing position that is and will affect many people and our world. I would like to read to you a letter that was just written by Uko Gorder, president of ACS, in response to our current situation. It looks long. It's big font with lots of spaces. It's not bad. It was on October 3rd, 1970, that the Nixon administration formed the National Oceanic and Atmospheric Administration, known as NOAA. Their stated concern at that time is the condition of our physical environment has intensified. It has become clear that we need to know more about the total environment, land, water, and air. Headquartered in Silver Springs, Maryland, this agency now conducts weather forecasting, monitoring the conditions of our oceans and atmosphere, manages and regulates fish stocks, and protects marine mammals within U.S. waters. Its scientific research and regulatory framework is vitally important to fishermen, farmers, maritime industry, and ultimately all of us. We all depend on clean air and oceans, real-time weather forecasting, i.e. tracking hurricanes, and safe and sustainable seafood on our dinner plates, and protect the oceans, resources, and environment. It is distressing to see the recent actions taken by the current administration, seemingly determined to dismantle and likely eliminate NOAA in the process. The ramifications are nothing short of disastrous in light of increasingly extreme climate-related weather events. It would also severely destabilize the fishing and farming industry and have a terrible negative effect on the U.S. economy. We ask all of you to lend your support for NOAA and urge you to contact your representative to stop further attempts to diminish NOAA's crucial services and to stop the layoffs of its workforce. We are deeply concerned by efforts to further undermine the critical Marine Mammal Protection Act and the Endangered Species Act, both also enacted by the Nixon administration. We hope you can join us to voice your concerns and contact your elected representatives, not only on behalf of the cetaceans, but for the overall environment on which we all depend. All right, now back to some fun things. On April 6th, well, the ACS San Diego chapter has a couple of fun events that we're going to be working on. Wanted to share them with you, so hopefully you can attend if you're willing and able. So on April 6th, in honor of Earth Day, we will have a table at the Rancho Bernardo Earth Day Fair being held at the Rancho Bernardo Community Park from 10 to 1. So please stop by and say hello. We've been doing this for several years, and it's just a great crowd, and it's wonderful to be there. And then our April 9th speaker series is a double treat. Both Dr. Barbara Taylor and Dr. Jay Barlow will be presenting, period. Barbara's topic is changing human behavior. Cases from around the globe reveal the need to expand conservation, need to expand conservation expertise. And J's is listening to beaked whales. What can they tell us about their behavior, distribution, and abundance? The April talk will be on board the Legacy. And then in May, the Legacy is going to be taken out of the water for some normal maintenance and repairs. So the May speaker, who we haven't quite nailed down yet, will be on Zoom, and we'll let you more about that. Then one other last note, if anyone is interested in helping out on any committees, just give one of us a nods up, or you can check that box on the sign-in sheet and we'll get back in touch with you. All right, so before I introduce our speaker, just a reminder to please turn off your mics and post questions in the chat so that we can respond at the end of the presentation. I was trying to reduce this, but your background is so extraordinary. So I'm just going to kind of rattle this off. Okay. Our speaker tonight leads the nutrition and veterinarian clinical research and diagnostic teams of the San Diego Zoo Wildlife Alliance. So you might be thinking, well, how does that fit into cetaceans, right? You think of the zoo and you don't think of cetaceans naturally. Well, the beginning of his extraordinary journey and career began with a veterinarian degree from the University of Gent in Belgium, a master's degree in marine biology from the University of Otago, New Zealand, a PhD in infectious diseases and marine mammals from the University of Florida. He gained much of his clinical experience during his time as a clinical veterinarian at SeaWorld. And it just keeps going. It is absolutely extraordinary. So I'm going to top off just a couple of things.
For such a young guy.
I know. You know, he has served as vice president of conservation medicine and science of the Pacific Marine Center, courtesy clinical assistant professor in marine mammal medicine at the University of Florida, co-authored over 60 peer-reviewed scientific papers, is a member of the Society for Marine Mammalogy, and is a former vice president of the International Association for Aquatic Animal Medicine and the former vice chair of NOAA Working Group for Marine Mammal Mortality Events. So I'd say it fits, right? His background fits. So please help me welcome Dr. Hendrik Nollens, Vice President, Wildlife Health, San Diego Wildlife Alliance, who will be presenting developing remotely collected health indicators for the endangered southern resident killer whales. All yours.
Thank you. Do we need to turn off any lights, you said, or are we good?
We do.
Okay. And then this is great. I don't have to introduce myself anymore. I wish you had shortened it. Oh, well, look at that. That works for me. I have to say, well, two things before I get started. I love this venue. This is an awesome venue to have these seminars. So I'll probably be back and bring my family. And then secondly, because you mentioned NOAA, so this project is, I don't do the project partners justice, but
Yes. A little louder.
a little louder. Okay. I don't, in my slides, I don't do the project partners justice. We are certainly not going at this alone, but NOAA is a key partner for this project. In fact, any of the field work, well, everything we do is under permit and authorized by NOAA, and we'll get into this. Actually, some of it is at the request of NOAA. Actually, a lot of it is at the request of NOAA. But also we work off of NOAA vessels with NOAA's lead biologist in the Northwest Fisheries Science Center. So them and then the Sea Dock Society and Wild Orca are sort of the key integral partners. So to your list of examples, this is the sort of initiative that would look very differently if we didn't have that support. Anyway, back to the slide. So I can skip my own introduction. This is great. Thank you. I'll start this here. As you mentioned, I figured there was going to be a question, the zoo and whales, how does that work? So, but at San Diego Zoo, we have organized our field conservation initiatives into 8, what we call them hubs, 8 hubs. There's sort of 8 ecoregions where we concentrate our efforts. It prevents us from being spread too thin and doing lots of things in lots of different countries. And it really is an intent to intentionally sort of focus our efforts into, so we're not like an oil slick, right? So we have more depth and actually can make some more impact that is longer lasting, hopefully. One of the hubs of the San Diego Zoo Wildlife Alliance is actually Oceans Hub. It's our second smallest hub of the 8 after Australia. Australia is our smallest hub. It has a number of species programs in it. One of them is heavily focused on polar bears. I'm pointing to Ann Bowles. I think she's over there. And then we have recently started engaging on sea otter, Southern California sea otter reintroductions, a long-standing tern and plover monitoring program. We are involved in banking of the now endangered sunflower sea star. So there really is, it's a budding initiative. And if you track the Oceans Hub at San Diego Zoo Wildlife Alliance, I anticipate that you'll see growth and expansion over the years. So the common themes within the hub across those programs are all our programs in the Oceans Hub are very focused and are entirely almost reliant on partnerships and alliances. There's nothing we do alone. There's a strong emphasis on promoting human-wildlife coexistence. We were talking about polar bears earlier. That's sort of where that most visibly is the area we're working on. Generally stabilizing declining populations. That's true for all of our hubs. But what's very unique about our Oceans Hub is the last bullet, is the amount of, because of the marine environment is a much more challenging or very differently challenging environment to work in than terrestrial environments. And so we very heavily engage our conservation technology team, our engineers, and use technology to accomplish some of our goals. So for this project, I first want to introduce you to the Salish Sea. You might hear that term throughout. This is, it's that body of water that's between Seattle there at the bottom of Puget Sound and Vancouver Island. It's sort of an inland water, but it's big enough that you don't feel like you're in inland water. You know, it's pretty open and exposed. It's the core of the habitat of southern resident killer whales. But they also go to open water for a good part of the year and are sometimes sighted in Southeast Alaska and sometimes sighted, at least some of them, as far south as Monterey. In that area, there actually are 3 killer whale ecotypes. I feel bad saying this to this group. You guys know all this, but the killer whales are not categorized into subspecies. They are ecotypes based on their culture, dialect, diets, habits. There are 3 in that area. There are the offshores, which we don't see because we don't go offshore. But what used to be called the transients and are now being renamed Biggs killer whales are actually now the predominant killer whale ecotype in that part of the world. And but we don't work on either of those, although we occasionally take advantage of when we run into transients to test some of our techniques, to get some experience, to get some samples or even data if relevant. The ecotype we focus on for this project is southern resident killer whales. They are salmon specialists, and as with other species, all species really that are if as a species you're highly specialized, you are much more vulnerable to changes in the environment, to threats, because anything changes and you don't have that breadth to start relying on other resources. So approximately 75 southern resident killer whales remaining and declining. We are effectively counting down by about 1 whale every year to date. The historically recognized threats for this population, so this is a very visible population. Sometimes you can see them from the waterfront in Seattle. People know there's a lot of awareness. There's a lot of community engagement. Historically, the threats to these animals that have been recognized has been prey, salmon, especially Chinook salmon availability. They rely primarily, 95% of their diet is Chinook salmon, and Chinook salmon itself has become endangered. Vessel disturbances was a big deal, was a big factor, and then contaminants, both legacy and there's some molecules that are being discharged today that people are still unclear on how big of a concern they are to these animals. But they are the second most contaminated killer whale population that we know of in the world. In recent years, though, these correlations between trends in the population and those threats, especially salmon availability, salmon and Chinook salmon abundance, has been weakening. There's something else going on. In the past, there's a really tight correlation in these trends. These days, that's no longer true. There are new threats that are being recognized or at least worried about. Some of them are changing sea surface temperatures. What does that do to the food chain? precipitation patterns, more intense, shorter duration range, freshwater runoff, and again, what does that do to the habitat quality and to the food that's available to them? But then also animal health. There have been several reports of animals that are in a compromised state and visibly compromised, and so that's what we focus on. For those of you who are active in conservation and conservation planning, you may be familiar with these diagrams. I'm not going to go into this too deep, but this is sort of a diagrammatic way of laying out. Conservation can be a very fuzzy, soft, squishy thing. You know, how do you define it? And so this is actually a method of planning and thinking and changing your approach when you're trying to maximize your conservation impact. So what this is on the right here in the green circle, without getting into too much of the details, you see our conservation target for this project are the southern resident killer whales. They experience they have a lot of old animals. We have animals in compromised health, decreased body condition. Some reports now, as of a couple of years ago, of inbreeding depression. They experience reduced food availability and obviously small population size, which is a risk by itself. On the left here, you see that those are the threats. Again, the ones that have historically been well recognized are the ecosystem threats, pollution, river dams that are interfering with the salmon spawning, some amount of overfishing, especially historically, noise pollution, and surface disturbance by by vessels. Now, these actually have all, to the extent that you can, are actually reasonably well managed. Some of them are more difficult to manage, right, to reverse and undo, but there's a lot of attention has been paid there. So the new threats that we're seeing is this changes in sea surface temperature, rainfall, and then disease. So if you look at this diagram, ultimately all these threats sort of converge, whether through bioaccumulation, food availability, exposure to pathogens, onto compromised health. And so that's where we place our intervention, because that's the purpose of this planning. If we work here, then that's how we maximize the impact of our work. What does that intervention look like? Don't worry about the small print. It's actually difficult to read anyway. But really sort of in steps is developing tools for measuring, assessing, evaluating health. Once we have that, getting the data to just start to understand what's normal from abnormal in a wild killer whale that's exposed to parasites and whatnot naturally, you know. Then once we have that understanding, we can actually make some assessment of individual animals' health, pod health, population health. And then that leads us to two potential actions. You know, this is sort of the how and sort of the stuff you must do to be able to do this, which is twofold. It's identifying opportunities and potentially performing emergency veterinary interventions. We'll get to that later. And then secondly is really using health or these metrics of health as a benchmark, a near-time indicator of how well the protective measures that are in place for these animals are working. When we work through that, our state of our target will improve. So first, let's talk about the how. This is, you know, how do we go about this? And so, rather than approaching this from a thinking of this when we conceive this project of, you know, if we were to examine a whale or an animal or pick a species in our care, these are the things we would do: draw a blood sample, do an ultrasound, you know, take a radiograph. We threw all that out, and we actually went out with the NOAA biologists and just started spending time with them and just to see what can we even measure from these animals, what can we see, what can we document, what can we collect. Start with that. And then from there, which of these things can we collect without the whales knowing? Because we don't want the whales to know that the doctor is in the house, you know, that the veterinarian is there. And then, but also then of those data points, which ones of those are actually useful, repeatable, and things that we would trust to make some sort of judgment of their health on. We started with over 50 potential metrics. We've whittled those down to about 30 now, and some of them are still under development, and we'll walk through some of these. One category of some of these health metrics are would be subjective qualitative observations. So there are those can be by myself to the extent or my colleagues on the boat to the extent that we're comfortable with the metric. But there are a group of biologists that we are working with very closely. They're an integral part of this work. Center for Whale Research, Wild Orca, who spends their, conditions permitting, and proximity of the whales permitting, spend every day on the water with those animals to do their work, to monitor boat activity, whatever their primary purpose may be. And they become very familiar with, they develop a strong intuition of what the behavior of the animals should look like, how they should service, how they should move, who should be with who should be close, who's usually far away, the males hanging there sort of in the wings. And so they become very, these opinions, these observed trained observer observations are actually very valuable, at least in some cases. And so we document these things. So these are biologists' opinions or reports of behavior, movement, social grouping, interaction between animals, swim speeds, etc. And in fact, Wild Orca, one of the project partners, has developed an app for collecting all these things. And so it's now being tested. We rolled it out to some other groups and actually are hoping to onboard the local whale watchers to contribute health observations into this database as well. The second set of observations is that observation still, but either objective, yes, no, or quantitative. And these include things like presence of skin lesion types, presence of injuries, extent of lesions of the portion of skin surface that is affected by some lesion type. These gray patches, or we have different definitions for some of these lesions by now. Or things like longest dive duration. What's the longest breath hold? Sometimes that's short for variable reasons, but if an animal has consistently shorter breath holds than it had historically, maybe there's a clue here, right? We can't make a decision or an assessment based on one metric. We have to put all of these together. But longest dive duration, respiratory rate at speed we just published a paper on that. This actually can be done from land. There's a group, Oceans Initiative, that does theodolite land-based observation. They measure at the same spot each time the whales come through, half faster swimming, half faster breathing. Great metric. It's like a fitness test. Put the animals on the treadmill and see if something changes. There's another group. This is not our work, so I certainly need to give them credit. That uses drones for body scoring, body condition scoring. So they have developed a method for using ratios of this width versus this width, and they can plot that over time, and they can see which animals are losing weight. The other thing they can tell actually is at least not with 100% accuracy, actually quite a bit below 100% accuracy, but at least occasionally is pregnancies. Turns out killer whales have a baby bump, and at about 1/3 of the pregnancy, the widest part of the whale actually shifts from before the dorsal fin to behind the dorsal fin, to immediately behind the dorsal fin. And actually, if you look closely in this photo, there's a little baby right there under mom. We can also get some biological samples. Here you go. I mentioned NOAA. This is Brad Hansen. He's our mission captain. Everything happens under his permit and usually from his vessel. There are two sample types we can get, and the first one is feces. We call them endangered feces because we think it's funny. But it actually has been a pretty amazing, the amount of things we have learned that we can measure in feces, and repeatedly, reliably measure in feces with some amount of variability because, you scoop it out of the seawater with a pool net. But things like indicators of bacterial flora, gram stains, presence of inflammatory cells, cytology, parasitology. We're working with Texas A&M Vet School to look at markers of GI function. We have some early indication that at least some of the southern residents have either anywhere on the spectrum from irritation to inflammation in their GI tract. But we don't yet understand for a whale that lives in the wild that has supposed to have some parasites in the GI tract, would be normal. Maybe that's normal. So that's one of these examples of things where we need to learn over time what all this means. hormones. We can detect pregnancy hormones in the feces, exposure to harmful algal toxins. And the one I skipped at the very top there is actually really, really neat. Now here at the zoo and at NOAA, we have assays where we can genetically trace back fecal samples to the whale. So we get a fecal sample, run it through the PCR, and it can tell us which whale this sample came from, which means that we can now take all that data and put it into the individual animal's medical record. We are developing a medical record system. So we are really getting to the animals are individually identifiable easily, at least by some, not by me. And so each animal has a record with repeated observations, so we can compare those animals against themselves. We are pretty good at getting feces, but we actually have get some strong assistance from this little dog called Iba. Iba is a trained, certified canine for conservation, and her job is to sniff whale poop. She is on the bow of the boat. Wild Orca, this is Wild Orca's dog, not ours, but Wild Orca is a key partner of this project. Eva is on the bow, and she is, she's got her goggles on and her life jacket, and so she will sniff for whale poop, and then gets excited and guides the boat to the sample, and the biologists can scoop them out, and then she gets to play tug of war. Yeah. Anyway, enough said. Lots of stories about. The second actual biological sample that we can get by now, it certainly was a challenge at first, is breath samples. At this point, only the vapor droplets. We're working on also trying to capture the gases that are exhaled. The breath, or at least the fluid that lines our lungs, at some point in its, as it makes its way through the lungs, is only one cell layer removed from the blood of the whale, for all of us mammals, and so it is to us the closest thing we will ever get to an actual blood sample from a wild killer whale. So we think we were very clever. We called it breath sample. Blow. The blood of wild whales. So we may one day submit a patent or a grant on blow. But anyway, so what we at this point in time we have, we'll get into this in a little bit. This has been very challenging to get that sample. Also, as you can imagine, this spray that you see on the slide is not all lung fluid. Majority of that is seawater. And so there's a little bit of lung fluid mixed in. And so we spent for the past 4 years optimizing the techniques for sample collection. We had a major breakthrough this past September. We'll show you that in a minute. And so we are now actually able to, we have enough material to start doing some respiratory health evaluation, which is important for cetaceans. Cetaceans are prone in general for developing pneumonia, lower respiratory infections. Not always clear if that's the primary insult, if that's the primary problem, or if it's something that comes on board in a compromised animals, you get bacterial overgrowth. But still, clinically, from a veterinary perspective, lung health is a very important part of cetacean health management. And there are some hormones, we haven't cracked that yet, but there are some hormones that you cannot detect in feces, but you can detect in breath. And so that's what we were hoping to analyze. So to get this done, we brought in somebody who was asking me about the conservation technology team. We have a department at San Diego Zoo Wildlife Alliance that focuses entirely on conservation tech, which is edge cameras, edge AI, sound traps, camera traps, you name it. These guys embraced this project, embraced the challenge. This is they started modeling the airflow around some of these annoying little drones that we see here now and then, the Mavics, and actually figured out where on the drone would you, so there's a propeller downwash, right? That's what this green and red is. But if you look at top, there's actually an area of negative flow right here in front where the air sort of gets sucked down. And so the drone, when it flies, actually also functions as a suction device. So it causes a downdraft, but that means that it pulls in air. And so that's where we position our sampling device. We've gone through a few iterations. Here, boys and their toys. They have a lot of fun doing this. Sometimes we go really low tech and then high tech. This is an iris that sits on top of these petri dishes with a remote servo motor that we can open and close if we want to. Turns out that actually complicated things too much, so we don't use that anymore. Anyway, point is, a lot of R&D happening and a lot of fun at the same time. So this is then what that looks like. So here we are somewhere in the Salish Sea. This is J-Pod, which is one of the 3 pods of Southern Residents. Here's our drone right there. This is the first rendition because it only had 1 Petri dish mounted on the drone, right? This is what the drone looks like. It's a small quadricopter. We have a 3D printed custom harness that has this Petri dish, which is the transparent thing on top there, right in that position of the downwash. It's angled to sort of get the maximum exposure. And so here this drone is tracking some whales. You see, there we go. For those of you who are out on boats a lot, which I think is most of you, you know, these conditions is what you get all the time, you know. Sea state is 0, no other vessels around interfering with your work, perfect backlighting of the breath, right? This is how it is all the time, especially in the Pacific Northwest. So we're behind the whale on our boat. There's the whale has just surfaced, exhaled. And so the way it works is the drone stays behind the whales. The whales come up, you know, you can see the black and white counter shading through the subsurface. We stay behind. When the whale surfaces, they exhale and inhale, and then they submerge. By the time they submerge is when we swoop in, and we, you'll see it here on this next slide. If we actually do our job right, you can see the circle, this halo, these little angel wings. For those of you who have watched Top Gun, that means that we actually hit the plume, and then we have vapor droplets on our plate, bring it back to the boat, process the plate, and do it again. Everybody loves videos, so I put one in here. This is what it looks like from the drones. So our drones have not just the Petri dish, but also have a 4K camera and actually a second infrared camera mounted side by side. We'll get into that a little bit. So you see we're behind the whale. Don't hit the water. And so swoop in there, get the breath, pull up. Looking around, where's the whale? Here you can see the saddle patch. You can see the genital patch. Wait for it, wait for it. This is actually really tricky. Our drone pilots tell me, actually the first time we did this flight, the first week we did these flights, at some point when they had their first hit, they looked at each other and they're like, we just invented the best video game ever. Because it really is a challenge. Southern Residents, for those of you who they tend to surface 3, maybe 4 times, and then they go on a long dive. When you go on a long dive, you don't know where they're going to show up. You know, you can keep the heading, you can keep the speed, but you know, they may change their mind, do something else. And so we have a little bit of a lag on Zoom here. But I forgot where I was going with this. Anyway, so this is a drone stays behind the whales, capture the breath, and go back home. Okay, so those are some of the metrics that are already in the portfolio that we do all the time. When we see Southern Residents, all these things we've talked about, we document all the time. This is our standard, our template, so we get a lot of data. There are a number of other things that we are continuing to look into, or you probably will for many more years. We are looking at using infrared cameras, whether it's handheld units like the one in the bottom right, or the ones that are mounted on our drone, to measure the breath plume. So the breath is warm when it leaves the body, right? It's brought up to body temperature. We're looking at if we can use some of these metrics that we can derive from it as another indicator of lung health or lung function, tidal volume. Challenging. It has just getting the volume of that plume has been a real challenge. Another application though of FLIR is to actually look at when you see a whale with some skin changes in here. If this is if this is a scar, it'll be cold. It'll just be the same temperatures as anywhere else. If this is an active skin change, if this is the product of a recent trauma, you'll have increased blood perfusion, which will show up warm, which you can see in some of those whales over there on the right. Also a little challenging here, which is why this is in R&D phase here. Cetaceans and killer whales, certainly on their dorsal fin, have these thermal windows. I feel awkward saying this in front of Anne because Anne knows a lot more about this than I do. But, you know, they have these certain windows that they tend to increase perfusion to when they want to dump heat, when they need to dump heat. And so it's normal to see some of these heat patches. So seeing one heat patch doesn't mean that there's a problem. The animal may just be thermally regulating. So again, we need these recurring observations. Something that we started looking into 2 years ago, and it was one of the primary focus of our field mission last September, is to actually use our high-resolution infrared camera from the drone to measure the intranasal temperature of the whales from up above. So this drone is about 50 feet up. The pilots have to fly right just in front of the whale and actually fly backwards so she can look down that blowhole, nasal tract of the whales. And so if they do it just right, which is the metric we use for actually getting the data point, you can see even though odontocetes only have one blowhole flap, they still have two nasal passages underneath. And so if you're lined up just right, you can actually see the two nasal passages and the septum, the division of the two nasal passages down there. So that's when we know we're aligned, and then that's when we take our measurements. Again, what we're getting at here is it started off as trying to see if we could measure the equivalent of a rectal temperature of the whales. Turns out that that's probably not it, but it actually could even be more. Preliminarily, it looks like this is actually more a reflection of that tidal of the exhalation function of the whale, which is almost more interesting and more useful than a core temperature, a rectal temperature would be.
Can you quantify that?
Yeah. Yes.
So you know that's 37 degrees Celsius or something.
So the way this is captured is not just like a picture on your phone. There's actually each of these pixels has the data embedded, and you can at the end of the day, we get back to the house, get back to the lab, we could download all that. And there's actually, which is one of the things that we need to do this September. There are a lot of environmental variables you need to correct for. It's reflectivity of the surface temperature, air temperature, humidity. There's a number of things that have changed that emission pattern. And so we need to correct for that. Anyway, there's a whole different conversation because we now will have some thermal standards on the boat for calibration next to every whale, et cetera, et cetera. We can't use a stethoscope, which is not actually that useful for cetaceans in general. But so instead, what we are doing is we use a directional microphone, not hydrophone, microphone to document the respiratory cycle, the sound, at least, the acoustic signature of the respiratory cycle of each southern resident killer whale each time we see them. We don't have any magic to say, this graph means this yet. Maybe an acoustician could help us with that, but what we want to do really again is be able to document a whale today and then again next year and again next year, and actually we're looking for changes. If something's changing, that could be an indicator of that something may be changing inside the lungs, right? Which is what we would want to know. With our new bigger drones and high resolution footage, another tool that we're looking into in the R&D phase, as everybody in the world, it seems like is AI, is actually, we're very actively working to have what the drone sees, we can see on a big monitor on the boat. It's mounted on the bow, and we are working to have machine learning to automatically have the computer tell us this is this whale, in the moving boxes J32, J36, but then also help our drones. If the pattern we were describing before, an animal shows up here, send the drone coordinates of where it should be because it's most likely to surface there again. So you're there in the right time, the right place to collect that breath. A lot of work needed here, but very exciting potential. So that's sort of the how. But who's our audience for all this? Like, and what are we making possible by doing this? So, big question. When we started talking about this idea of health assessments for killer whales, well, first of all, people thought we were a little crazy. It was it's like, hey, what are you talking about? Wild killer whales? How are you gonna do this? You can't catch them. You're not allowed to approach them. You can't harass them. All these things. And we could have not tried, but we did. But we really set out thinking that what NOAA specifically would want from us is information that is much more real time on how the whales are doing today. So the two metrics up until this work that NOAA has available for informing measures to protect southern resident killer whales are head counts. Every year Center for Whale Research does a census, and the body condition scoring that the other team was doing. Now, for a small population of 75 whales that are animals that are long-lived have long gestations, 15 months. There's a lot of stochasticity in that data. You're going to be waiting for years to recognize that there's actually a trend that's positive or negative. Well, we know it's negative, but to start to see a change in that trend. Body condition is great for some reason, but that's the first thing you do when you go to the doctor. You stand on the scale, and then there's everything else. Right? It doesn't give you any information on what's going on. It doesn't. Not all health states result in body loss. Not all body condition loss is because of a disease problem. Right? So it's really it's an important piece of information, but out of context, it's not really that useful. So we thought we were going to mainly the main value to the resource managers of this work is to tell them, how are the whales doing today? How many whales are out there today that we're concerned about? How are the youngsters doing? How are they transitioning from weaning to, from nursing to weaning? And that use, that interest is certainly there. But what NOAA really drove us to is these the veterinary interventions, which I thought was sort of like the most far-fetched applications, going into this. But no, this is what NOAA wants, and there are actually some precedents that has allowed for that thinking today, and we'll get into some of that in a little bit. What these veterinary interventions could look like. This could be disease outbreak prevention. This is a paper that was done by the folks at the Center for Whale Research. You guys probably know Ken Backham and his team on what a morbillivirus outbreak would look like for southern residents. Fortunately, we haven't had to experience southern morbilli in the Eastern Pacific, but just imagine if you did. This topic came up some years ago, 4 years ago now, around COVID, because there was a SARS-CoV-2 concern about cetaceans. And then again, now 2 years ago, it's still today, avian influenza. It's been found in harbor seals up there. What would it do if it gets to this population? If we decide that's useful, having these platforms, knowing the whales, understanding their health states, having veterinarians embedded with these on-water teams actually allows us to at least have the possibility to immunize, to vaccinate all the young females, or whatever it may be, the individual whales that are most valuable for population recovery and sustainability. But this specifically is what NOAA has been interested in. So this project, we had been in a conceptual phase for a little bit, but I think it's 5 years ago now, I lost track. There was a young 4-year-old female, J50, who showed up in summer, emaciated, skinny, and started progressively emaciated over the course of summer. There were members of the public were calling for intervention. Some were saying, leave her alone, don't you dare come near her. NOAA was really in a tough spot here. There was no winning for them. Ultimately, there were town halls, there were public hearings, there was a lot of hoopla, and ultimately J50 just disappeared. She died. That really was the accelerator for this project, because there had been this talk of these veterinary interventions. I should say, at some point in that process, a few of us were asked to do something. And so we went up there and we actually delivered some long-acting antibiotic to J50, but it was too little, too late. It was a Hail Mary. We didn't know what we were potentially even treating in this population. So it was, with hindsight, it might actually not be the drug anymore that we would choose today, the first go-to drug based on what we've learned. So that was the accelerator. So fast forward to, again, the years are getting blurry. I think it was last September. We were on the water and behind J-Pod, and we've all smelled, I think most of you have smelled whale breath. It doesn't always smell great. Mm-hmm. This was something else. This was like necrosis, just like putrid smell behind this group of animals. There's nothing good or normal that could smell like that. And so a little bit difficult because it's a traveling group, but we were able to sort of triangulate out which whale we thought it was. We thought it was J32. Launched a drone, put the plate on top, actually flew in, followed her, successfully collected some vapor droplets from J32, brought it back to the boat in the lab, and we're actually able to see that we saw abnormal cells. We actually had red blood cells in her breath, which are not supposed to be there. Mm-hmm. So big success. We, for the first time, were able to say, it's this whale has something abnormal going on based on, you know, our sensory perception, but also based on this piece of lab diagnostic piece. Also, her blowhole temperature was 4 degrees Fahrenheit lower than the intranasal blowhole temperature of the other whales in the pot. So something was going on. Her core temperature hadn't dropped by 4 degrees. That would not be biologically likely, but something is changing in this airflow, in that air mixing. So we felt reasonably comfortable saying that we thought J32 was dealing with something, we don't know what, but something upper respiratory or at least respiratory. We reported this out to the resource managers. We saw J32 the next day, same result, more conversation. What do we need to do? She seems fine. Her calf is skinny. We saw her again, I think it was 3 days later, and the breath smelled normal. Lots were drawn, got breath plates. We did not see abnormal cells. We didn't do a thing, right? It's not, nothing was off because of what we did, but it is the first time that we were able to, that somebody was able to actually respond to a potentially compromised whale in real time. Luckily, we didn't have to do anything, but it gives you an idea. This is something, this is also what it could look like. Different species, don't need to explain that to you. This is North Atlantic right whale. Shortly after this J50 story, like literally within months, this calf, I'm trying to, mom's name was, it was something that meant right in or anyway, so I forgot, Dextera or something. So this is a calf, and when this calf was 5 days old, it was struck by a ship right off the coast of Jacksonville. And so this was a propeller mark right across the head of the whale. And as you know, that's really sort of the upper lip and into the mouth of that animal. Again, Noah asked, what can you do? What can you do? And so here we had a clear diagnosis. This was trauma. You know, we can't go close this. We can't go catch this animal. We can't go bring it in. We can't bring it to a rescue center. Surgically, there's nothing you can do for here. The best thing we can do is try to make sure is to give the animal time to hopefully heal and make sure she doesn't develop an infection and become septic in the meantime. So again, we deployed just like we did with J50, and let me see, Derecha, that was her name. There you go, not Dexter. And so we deployed. We spent a few days tracking these whales up and down the coast. We had a spotter plane from Georgia DNR, from Florida Fish and Wildlife, all looking for mom and calf. At some point, we found her, and this is our intervention vessel. This is a fish and wildlife biologist who's used to darting right whales, so he's used to crossbow usually. This is a custom rifle that was built 20 or so years ago for actually tranquilizing right whales at sea before disentanglement attempts. It's only ever been used twice because it's really hard. All the stars have to align. We now have a system like that here on the East Coast at the Pacific Marine Mammal Center, so this would be possible. This dart has a 57 ml injection volume, which is huge, a huge volume. You know, usually you get like 7.5 ml in a dart. We packed it with long-acting antibiotics. So we were approaching the whale. We're like, Jamie, you didn't shoot. And the plane calls back to the boat. Mom's on the outside, calf between mom and the boat because the dart actually has a tether and we don't want to shoot over top of mom. Mm-hmm. The tether is there so we can retrieve the dart so that no whale shows up with, with a harpoon in its back somewhere. And so here we go. Calf surfaces. Oh, we missed a shot on Zoom, didn't we? With these frames skipping, we missed a shot, but you actually see, you would have seen that the dart goes in, you count to 7, retrieve the dart. Clearly there's actually less of a reaction than I thought there would have been. There was a change of course, and here you would see that mom and calf are now picking up speed and taking off. This story did not end well. We learned from our imagery that same day that this calf actually at some point was swimming and must have nursed from mom, and there was a milk plume behind the calf as it was swimming, so that it had cut too deep into the lip. The animal wasn't able to. It was able to nurse, but it wasn't able to hold it in its mouth, and so calf was never seen again. But this is again an important precedent to have to have a government agency to ask you to, yes, go do something, to have the public support, to have the scientific community support, to have whale watching support, all the cetacean stakeholders in support, at least largely, for doing this work. This really is sort of setting the precedence for what we might be doing might be able to do for southern residents going forward. How am I doing on time? Wow, we're talking for a long time. My last two slides here. I don't know if that's good news or bad news, but so this is where we are today. Obviously, there's a lot more detail and a lot more granularity in all the work we do. We can get to any and all of it and into more detail if you want to. But where we are at really is at this point is so we have to continue to do work, continue to do local capacity building, continue to promote these collaborations. We actually have a lot of buy-in now, which is hard to get to, for data sharing between the killer whale biologists. It's really challenging because these data is grants, is revenue, is sustainability for your program. And if you start sharing that, how does that, does that compromise your own program? Well, we have agreements now. Things are going really well. And where we're at and what we are working towards now is really to start taking this effort from twice a year, September and June is when we go up to do different parts of the work, but to really deploy that as a local year-round effort. The biologists, professional whale watch operators, other local teams, have them collect samples, have them collect data, at least those data or sample collections that we trust, that we know won't interfere with the whales or potentially go wrong. And so that's where we're at, is taking these 20 or so metrics that are up and running, that are working, that we trust, and just get the community who's on the water with these whales year round to get those data and collect them, enter them into these iPads, so it goes into the database. With that, this is where I should have thanked my project partners, but I always forget to do that. But I think I did it a few times. We have NOAA, obviously, Wild Orca, and CDOC, and Wild Orca and CDOC just merged into one entity. So really, this is at this point a three-way alliance between us and NOAA and CDOC Society, which is UC Davis field office up on Orcas Island. So with that,
Good.
I will go ahead and take some questions.
Thank you. And thank your partners.
Yes. Thank you, partners.
Since we're recording it, you get to get credit for thanking your partners.
Exactly.
We have a couple of I think here we go. We got a couple of questions, and then I'm sure we have questions inside our group. So
One moment.
how solid are the whale feces, and do you feel the samples collected near the surface are represented?
Oh, great question. Somebody's getting to, someone needs to come join the project. So
that's George, one of our board members.
Okay, yeah. So the feces vary. And so sometimes if we're really lucky, we get a little clump, but a lot of times it's like a raft of feces that floats to the surface. What's interesting is that when you look at any of the other ecotypes of killer whales, that's not what their feces look like. In fact, if you were at SeaWorld and whale poops and a raft of feces flew to the surface, they'll call you in. Something's wrong. But in southern resident, that is certainly very common. But we don't know. I don't know what the bias is, and because it could be that the feces that float to the surface are the ones that are abnormal, which is why we're seeing evidence of irritation and inflammation. And we know that we are sometimes missing feces because we can see whales foraging, or like Maya or others who are working on the project. They see the whales foraging all day, and there's no defecation stuff from what they can tell. So on that day, they're probably diffusing or sinking. There may be a bias there. One way, actually, we just, again, our engineers at Conservation Tech Lab just acquired a device that's really cool. It's from our drone. It sounds very James Bond. There's this little collection device that you can lower from the drone on a tether, so you can fly over the whale from as far as you want. You just watch for defecation, and then you hold stationary and you lower the bucket, and it submerges, and then you can. It's a remote trigger. You can open it, and so we have seen defecations happen. And then, we'll put a marker, or the drone stays overhead, the other boat comes in, try to scoop it up, and there's no feces. It's all sunk out. So having that collection device on the drone will hopefully help us. We're going to have to figure out we'll hopefully answer some of that bias. We may have some challenges to get that bucket right into that feces. We're experimenting at the park with a little plankton net that is weighted that you can lower from the drone instead and sort of drag it to the water. And we're learning what does that do for flying the drone. So it's more like a sieve that you're pulling through the water. But that would get at that bias. It's a
So
great
for
question.
control, both for the feces and for the respiration, you take samples of seawater away
Yes.
from it because you'd have to cancel out whatever is naturally in the water.
Yeah, so we have to subtract the microbial community that's in the water, right?
Yeah.
And but also if you look at the bacterial IDs that you get, you can just sort of have an idea of what's potentially pathogen, what's seawater
Well, the
contamination.
RBCs in that respiratory sample, that's not
that's
gonna
real.
typically happen in the water.
Another challenge is that we find some bacteria very commonly, but they are actually coming from salmon. They're salmon gut bacteria, so that's sort of a third origin.
Yeah.
But and then for the breath, the way we actually, so with the feces, you got feces, right? It's there. We do SNP genotyping. With the breath, the way we confirm that we actually have lung fluid in our sample and how much is through a PCR that detects host mitochondrial DNA. So we get a plate back to the boat. The first thing you measure is there killer whale DNA in this, or did we get air or seawater? And so we can calibrate our breath samples based on just generic killer whale host DNA presence in the sample. So that's our calibration for the breath sample.
And then Laura Keener is asking, has anyone looked at the gut microme? It might be a setup. I see you smiling. And would it be useful? So
the gut, which, sorry?
So it was, has anyone looked at the gut microbiome?
Yeah.
And if so, is it useful?
Our NOAA partners have, and our NOAA partners have it could be useful, but not yet. Actually, and with any of the species that Laura helps care for, is we have the same problem. You get this dendrogram of bacteria. And what does it mean? There's actually a guy here at ACS-SD who's sort of a leading authority on using microbiomes in a clinical context, and instead of the actual names of the bacteria, they have a way to 3D graph changes in microbiome, which is
Cool.
Clinically much more interesting, because I don't need to know what bacteria are and what abundance in your GI, but if something suddenly has shifted, I need to understand sort of what's your normal variation on your day-to-day, and then suddenly you're out here, well, something happens. What it's and then you can go say, okay, what else is going on with this animal? Is that the only thing that's changed, or is it also losing weight and it's no longer defecating because maybe it's not foraging, and that sort of thing.
My own research was in respiration, so I'm quite interested in the tidal volume.
Yeah.
So are you calculating that tidal volume from the flow rate? Just kind of going backwards?
We haven't been able to calculate the tidal volume because it's really hard to draw a boundary on that.
Yeah.
It's actually that imager we used it's a camera, I forgot what it's called, but it overlays the heat signature with a CO2 signature.
It's a very difficult measurement to take, and we did the stovepipes over the top of Gigi when Gigi was in SeaWorld.
Yeah.
Back when I was used to at the UCSD.
So what we're looking at with the infrared imagery, actually more so from the drone now than from the from the boat-based camera, is to look at the rate of temperature increase or decrease, and to see if that can be a metric. If we can measure that, then we can go do a study to see if that's correlated with effort, with respiratory effort and as a metric for, it's sort of like the ejection speed instead of your actual tidal volume.
Yeah, interesting. How about in-house? Here we go. And did repeat the question for the people.
Okay, so yeah.
String of microplastics in the bottom.
Yeah, so I didn't put it on here, but our the fecal samples come to us here in San Diego for analysis. It's actually our reproductive sciences lab that does the genotyping, and they measure for hormones. But two other things they measure is microplastics, and then PCBs, I believe, not DDTs. But we don't measure that in-house. It goes to SDSU, and we work with Una Han, I think is her name, is the lab. They are a high-end sort of spectrophotometer lab, and so we are measuring it. But the hard part with microplastics is the clinical interpretation. You can find it. Is what does it mean to find? That's the hard part, right? And so there's a lot of things we can look for, but we're trying to. We have a limited amount of time with each whale, a limited amount of sample. You know, that breath sample is like microliters, those vapor droplets or the feces. It could be 500 ml, or it could be like, you know, a fingernail full. And so we really try to focus on what could give us an indicator of the health of that animal. And with microplastics is really hard to interpret from a clinical perspective. But we measure it because we can, but it's not part of our matrix.
Finding,
Yeah. Oh, yeah. Yeah.
but
Yeah. Not me,
not to
but
say
yeah.
you're alarmed or.
No, not yet. No. I mean, you can find them everywhere.
Question, are the salmon contaminated that they're eating?
I don't know the answer to, they may be, I don't know. Sorry, the question is, are the salmon that they're eating contaminated? I need to remember to repeat the question.
Yeah.
I don't know the answer to that question if they're contaminated. I mean, they live in the same environment, so I'm sure they absorb what is in the environment. They don't bioaccumulate like killer whales do, right? Because a lot of pollutants love fat, or they get absorbed into fat. And so, but salmon has fat, not as much as a whale. But the salmon is endangered not because of pollution. It's actually because of urban development. Their life history, they go into freshwater, brackish water and freshwater habitat to spawn. You know, and there's waterfront property is popular, and then, you know, you put a seawall to protect your property, and then on top of that, you know, when they go upriver, there's hydropower, which is good from a footprint point of view, but not for the salmon. And so it's actually all that urban development has interfered with the reproduction of the Chinook salmon. And actually, it's just a few months ago, the Klamath River dams came down in Northern California. And that actually historically was one of the river mouths that southern residents used to go to feed. And so it's very exciting that there were immediately Chinook salmon running through there again. And so that's only one dam, but it's hopefully it'll help.
So a lot of contaminants are just right in their water there.
Yeah, so I'm not the contaminant expert on the project, but I'll tell you what I know. The main concerns for killer whales are PCBs, DDTs, and fire retardants too. The things that you see find in pillows, the stuffing of pillows. PCBs, DDTs have been just like in LA, right? You know, here a dump site right here. PCBs, DDTs are not used anymore commercially, but they're legacy, so they persist in the environment, and actually we know they persist. They settle into the marine sediment, and any disturbance of the marine sediment through dredging or storms or otherwise actually stirs all that up, and now it gets taken back up into the food chain and ultimately bioaccumulates into the whales. The pollutants with the southern residents is actually an interesting question, because the other most contaminated southern resident killer whale ecotype are the northern residents who live in Canada, culturally very similar. Sometimes they interact, they never interbreed, but they're breeding just fine. And so that sort of would suggest that maybe not. But also there's an important piece of data missing. In the last, I think it's 10 years now, there have been no more biopsies collected from southern resident killer whales, because an animal died shortly after a biopsy, and sort of it became taboo. And so we don't have any data on this segment of killer whales over the last 10 years to see if they are still building up pollutants, or if that's a problem that's sort of grown out of the population.
I just remember, because I was up there looking at the whales, and I just remember that there's a low survival rate of the calf because there's so much toxins in the mother's milk that the babies can't really make it very easily.
Yeah, that was clear. That is actually something that was very, I don't remember the paper, but a pretty clear link that was flagged with North Atlantic right whales first. And then later there was a pollutant milk transfer study done at SeaWorld that actually showed, so you could put the coefficients of how much pollution that goes into food actually ends up in the milk and then the blood of the calf. So it is a concern for sure. We do see this is my subjective interpretation of our observations to date, but the lack of recovery of southern resident killer whales actually isn't necessarily about mortality of animals. And it's actually, and again, I'm on a limb here a little bit, but just speaking as a clinician, right? I don't have all the data. This is my veterinary intuition. The problem doesn't seem to be cycling or conceiving or maintaining pregnancies. There's a real hurdle with these calves around about 30 days. Of those, there are calves that we entirely miss, that we never see because they're born, and, you know, a human never sees them. But we have in the last 11 years, there are 6 or 7 calves that have a pretty much a closely known age, you know, within a few days, because they would still have the umbilicus or something when they're first seen, that die around that 4 to 5 week mark. And just imagine a population of 75 animals, if you could have 5 more youngsters in just 10 years. I mean, that's a crux for your recovery right there, right? Yeah, and then the question is why. But so this is the point of the project, right? You're embedded in the community, you get the data, you get all the insights and the things that don't go published, and you know, because you're looped in, you're there with them on the boat talking to them, and so I feel like we're really sort of honing down this problem onto at least the health side, the veterinary side, onto these youngsters, not around weaning, around that post-colostrum phase.
Blubber, they get into the whale's blubber, and then of course goes through their milk and.
So yes, and so yeah, the reason I brought that up was actually exactly that, you know, the question of pollutants. So that would fit with a pollutant transfer from mom. If you were to get that, the ones that are most at risk is actually the first calf, because let's say a female killer whale on average has her first calf, let's say at about 12 years of age. You know, she's been eating for 12 years and accumulating, you know, toxins for 12 years. So that first calf gets 12 years worth of gunk. You know, every calf after that, let's say it's every 4 years, gets far less, right? Progressively less. And so your survival should go up if you're a calf. But there's confounding factors. You also have a more experienced mom. You know, there's stronger moms. Other things have changed over decades. And so it's hard to tease out.
One of the things that I'd like to do, first, thank you very, very much, but I was.
Oh, you brought my beer out.
I left it out of the screen, so.
Yeah.
Well, my wife was born in Victoria, and I got to meet her uncle, who was a commercial fisherman, and in my lifetime, the killer whales went from coyotes that were being shot, coyotes of the sea. So when I met him, my first introduction to anything about orca was they didn't like that, and they just did everything they could, illegal stuff.
Yep.
But my point being is the awareness that SeaWorld and research and NOAA has brought to this point where you're standing here telling us how much effort is going in to try to help save 75 whales. I mean, it brings a tear to my eye, you know. In my lifetime. I think that's just really, really cool. And I want to emphasize what Jane started off with the letter, and Noah is the, you know, and you could comment on this, but is the disaster of a loss of 5 years or 10 years worth of the ability to gather data as one administration changes and maybe to a more favorable administration. But you already talked a little bit about we don't have data during a really critical point. And that made me think about, as funding gets cut, you know, I mean, you know, the average person probably doesn't understand how important that is, but.
Yeah, and even if funding is only cut for 4 years, let's say, the amount of time it'll take to rebuild
To get it
and
going.
become a trusted employer, all the expertise you've lost because they've moved to work somewhere else and not going back to a bad employer.
Yeah.
Yeah, I'm at a loss, just like.
Yeah, that's, I mean, that's, we
And that's
started
just
with
NOAA.
that.
That's
Might be a
just
good place to
NOAA,
end with that, so.
Fish and Wildlife, DEA, USDA.
No more other questions?
No, that's, thank you very much.
And had one. All right, yeah, I'll save it for later.
We want to, oh,
Oh,
um,
are we still on?
a bit about how you're getting information about these calves during that tricky early life period. Because it seems to me like there's maybe some behavior questions involved in all of this.
Yes, yeah.
Are still, I haven't seen anybody try to get at that yet.
We will try to get at that because this is sort of a recent realization, at least of ours, that window is a critical window, and there's a questionnaire we're putting together. So these data are there's a questionnaire we're putting together that actually captures some of that. Has mom? Who is mom? Is she an experienced mom? Has she ever even seen a calf being born in this spot? Does she know what these things are? You know, the questions that we. But then.
Her mother and the rest of the cohort out there, because that's gonna be, I mean, in.
Males in the pod. What were the males doing? Yeah, that's where actually Megan Owen, we were talking about her, is joining the project this September, and so she will be she will be our behavioral ecologist on the team moving forward.
And Laura's throwing you another one as we go out the door here, and that is
Laura.
how quickly is the population decreasing? I may have missed this.
It's about one whale a year. We're counting down by about one whale every year. It's not quite that, luckily, and actually sort of in the last couple of years, in the last 4 years, when we've been doing this health work in earnest, one of our strategies for validating some of these metrics is actually when a whale is clearly compromised and on its way to dying, because that's really an opportunity for us to validate some of our metrics, even if we can't do anything for that animal, to have a southern resident that is clearly compromised because of loss of body condition or otherwise, and to be able to get data from that animal and compare that to these other data is really will help with recognizing or confirm that we're recognizing normal from abnormal. But we actually haven't had that in the last few years, which is a good thing, which is a great thing.
Yeah.
But but we have had, we were up in it was in September, we were up there and El Pod, a calf was born. We actually, because we were there and she stayed, the southern residents left, she stayed behind inshore with her calf, probably because it was safer, away from males. Although now we have these transients that we don't really understand what they would do, but.
Very slowly researched.
Yeah. So, but we followed mom and calf. We had we were up there for 3 weeks, and we had, I think it was 3 encounters with her, because we were able to figure out her pattern of how she was going around the island. So we were able to meet her twice after the first time we ran into her. And so the calf looked strong for those 3 weeks, and then 2 weeks later, it was found, it was swimming, flailing at the surface, and was missing.
Ouch.
So. But so behavioral piece, so we get we put a lot of emphasis on, we just, when there are calves, we just put the drone up, put it up high, do nothing else but film. We don't know yet what we're looking for, but at some point, we're going to want to go back to that. There you go. Yeah.
Okay.
Yeah.
Enough said. What's the sex ratio? Do you know all 75 individuals?
I don't, but yeah, they are known. Yeah, you can look it up. There's a photo ID catalog, and at least going back to the last 30 years, the genealogy is known. And in fact, the full genomes have been sequenced from each of these whales. We know the paternity, which is where that inbreeding depression paper came from. So yeah, everybody is known. Everybody has a known gender. Sometimes young whales change gender once because, they got it wrong at first.
So,
But
it's section.
I don't know it off the top of my head, but what we do track is the number of females with reproductive potential, and that's 16. It's something like that. That's the portion of the population that could actually contribute to recovery of reproductive age.
Breeding, potential breeding.
I might be 18, but it's something in that order, in that range. Yeah.
That seems low to me.
Yeah. It is a very top-heavy population. There's a old, the pyramid is upside down. The age pyramid is upside down. So,
That's gonna be like 20%, 22%.
I'm not good with math.
but I don't know the male versus female.
18 out of 75 is probably gonna be 23%. And if you think that they're taking kind of an every other year approach, right? So essentially you got almost 40% of the
And then
females that are either
5 or 6
having a
males.
baby or trying to have a baby, right? So they're a reproductive, 40% of the population. I mean, I think we could get that to grow.
Well,
We-
then, but losing 5 or 6, was that last season? I mean, 5 or 6 were lost.
Over the last 11 years, I think it was, yeah.
Oh,
Yeah,
over an
but
11
so, okay,
year.
but to get to your question, now I know where you're getting at. So we actually published, I think it was last year, a PVA, a population viability analysis, where we looked at the exact, not me, but Rob Williams, Oceans Institute, and we all contributed, but modeled out what is currently in the population, the reproductive potential, the anticipated reproductive output based on historical data. And at the current, the graphs are really sort of depressing. At the current rate, the population is gone is functionally extinct within 50 years, I think it is, which is 50 years because they're so long lived. But if with the current measures in place and veterinary intervention, he counted, if we can improve the health outcomes, so the survival of one whale per generation, we actually significantly improve the chance of recovering southern resident killer whale population.
That's good. That's huge.
Yeah, you'd actually could go to 1% population growth on an annual. They could achieve a 1% population growth annually.
Even with the inbreeding, the
Yeah.
genetic bottleneck.
Inbreeding is not a good thing, but inbreeding is something that we think of as a threat amplifier. It makes things more difficult, but it is rare that you have to have really. It is rare that inbreeding is actually a primary cause of mortality, and so it makes things more difficult because you're genetically and immunologically or otherwise less diverse, right? You have less genetic diversity in your biodiversity toolkit, if you wish. So you have less tools to work with, but it does not make it impossible. I mean, if you look at California condors, northern elephant seals, I mean, there are a lot of examples of species that have recovered from small numbers. Cheetahs,
I like the
they have one genome, but there's several cheetahs have one genome, but there's now hundreds of cheetahs again.
white whales. I was astounded how low the numbers, but
350 or
but
something.
they didn't find any, the gal, the researcher that did the talk last year. What did she say the numbers were?
She's talking about the North Pacific right well.
Oh, North Pacific. Oh, yeah. Less than 30. North Pacific is less than 30. Mhm.
But they found nothing genetically bad? Did I say that wrong in the talk?
Well, they don't have very many reproductive female on it.
Yeah.
Which is the bigger one.
She's so positive,
to
and I was crying with the numbers.
I'm not showing any signs of having serious genetic defect.
Yeah, that was stop short of me.
Well,
So-
they live.
Yeah, and there are two pieces here, right? You need to have females to breed, right?
Right.
You only need one male, but you need to have, your
Right.
females are sort of the growth limiting factor. So there's that. There's definitely that's a piece. But it's not just about head count. I'm trying to channel Oliver Ryder here, our conservation geneticist. It is. The numbers matter, but what's even more important is actually the amount of genetic diversity that is still encapsulated in the population that's remaining. If you can have, I don't remember, his analogy was somewhere with vaquita, but I forgot exactly how it went. But basically, you can have species in small numbers that have genetically more recovery potential than species in higher numbers. Obviously, there's sort of a reality of you need to have enough females, to carry the calves, but 30 whales could carry more genetic diversity than 75. I don't know if that's true, but they could.
My head, there was like a number of 50 or something. I remember when I was there. You have to have at least 50 pairs to, genetically stable. I mean, I'm trying to think of the words I'm trying to say, but I guess that's wrong then.
Oh, I don't know what. So I'm not a population modeler, so I'm not going to say anything wrong.
Like a
but-
bottleneck effect. You
Yeah.
think of how many marine mammals have been hunted to near extinction to become uneconomically feasible to get the last ones. But they're down to really limited numbers because of hunting, and they pop back.
Mhm.
And you think about what that bottleneck effect you know, the Adam and Eve effect or whatever, but typically we refer to it as a bottleneck
Right,
effect.
right.
And they come back to be robust populations when they had a really pretty limited gene pool
Yeah.
to recover from. So it's interesting. Thank you. We will close it up and we'll continue to ask questions.
Yeah,
Let's
okay.
close up
Very
the
much.
Zoom.
Bye, Laura.
Just,
Uh,
hang on, don't turn it off quite
Oh,
yet.
okay, so
And let's let Jane,
Hendrik,
and
that was
we-
great.
yeah,
Nice to see
You
you.
can,
Hey,
you can open
Jane.
that, this is a reward for
This is
Next
behavioral.
time.
positive reinforcement.
Yeah, positive reinforcement. Go ahead, Jane.
Good night, everybody. Thank you so much. It was a wonderful presentation and we look forward to seeing you next month. Have a good night.
Can I add something?
Sure.
Okay,